Hi, my name is Mia Wallach. I'm a candidate for a bachelor's degree in business administration at Northeastern University. Today, I will be discussing my research project through the CaNCURE program on PARP and CDK inhibitors for cancer therapy. In this experiment, my colleague and I investigated the synergy between two chemotherapies, dinaciclib and talazoparib, when administered to cancer cells in combination. Talazoparib blocks PARP1, an enzyme involved in DNA repair. Dinaciclib inhibits cyclin-dependent kinases, enzymes that are key to cell cycle regulation. In combination, they are able to produce synergy. Encapsulating the therapies in nanoparticles enables more effective delivery to the cancer tissues and decreased toxicity to the rest of the body. In order to conduct this research, I performed the following techniques: nanoparticle synthesis and characterization by size, surface charge, and polydispersity, cell propagation, performing series dilution and combination treatments on cancer cells, MTS assays to assess synergy, and fluorescent microscopy to confirm the drugs entered the intracellular fluid and nucleus of the cancer cells. By filming a lab technique class that was hosted in my laboratory, I also learned about gold nanoparticle synthesis and top-down nanoparticle synthesis. We conducted extensive testing in the Calu-6 human lung cancer cell line. The results initially looked promising, but after normalizing the data to untreated cells, little synergy was demonstrated between the two chemotherapies. We then changed direction, testing in the 41 mouse breast cancer cell line. In normalized data, the combination therapy of the nanoformulations produced lower cell viability than either therapy alone. We also conducted testing in the 3666 mouse ovarian cancer cell line. This cell too demonstrated lower cell viability for the combination therapy than for either therapy alone. More work on this research project is still needed. In the future, we hope to further investigate the optimal cell line and dosage in vitro. We then plan to conduct an in vivo portion of the study in order to investigate cytotoxicity, tolerated dose, pharmacokinetics, and therapeutic effects. I learned much more than just wet lab techniques from my CaNCURE experience. I also learned how to change direction in a project when data suggests poor results, how to write a research proposal in accordance with the Food and Drug Administration, and how to work with a small laboratory team. Most significantly, as an aspiring future physician, I learned the vital importance of continued innovation in oncology research. I'd like to thank Professor Sridhar for being my CaNCURE mentor and for offering me invaluable feedback on my projects, as well as for organizing the CaNCURE program. I'd like to thank Professor Vandeven for the countless hours she spent organizing the CaNCURE program and for the esteemed professionals she procured to speak with us. I'd like to thank Shichong Yang, my day-to-day mentor and co-investigator, for his endless patience and curiosity and for welcoming me into his research project with open arms. Lastly, I would like to thank the National Institute of Health for its generous grant that made this experience possible. Thank you.